of Horticultural Research, Bangalore, and his topic is going to be molecular approaches for the horticultural crop improvement.
Everyone, hope I am not disturbing your sleep. Anyway, it's on later vein. Friends, in fact, it is very nice to be an horticulturist when all the crop breeders are here. I am very proud to say that the horticulture, which is a premier product, has surpassed the agriculture production. It's around 286 million tons now. See, way back in 1950, it was around 30-35 million tons. Now we have crossed almost 286 million tons. So I am really proud to say that, and this credit should go to the breeders, the entomologists, pathologists, horticulturists, and the farmers. Probably deliberately, I did not mention the biotechnologists because the biotechnologist role will be year afterwards. I know for last 20 years, a lot of work is going on in biotechnology. Now, as a head of biotechnology for last 5 years, a lot of thrown from the ICR that we have given millions of rupees. What is the products which has come out? In fact, it was very difficult to say that, yes, all the markers will not give product, all the technologies will not give product, but it was very difficult to convince the ICR also that the biotechnology will not give a product very shortly. But after seeing this, probably you will decide that definitely we'll have a better products in another 10 years. Recently, in fact, we have formulated a Vision 2050 where we have to produce around 530 million tons of horticulture produce during 2050. And already you know that the breeders have already reached the plateau, and it is now the biotechnologist has to help the breeders or horticulturists to go for this. So with this, I start my lecture on the use of molecular markers in horticulture crops. Probably, if you see, the basic knowledge of structural and functional is very, very important for understanding of a crop. This is where actually, in fact, we did not go initially because we went, whatever somebody has did, we went, we just copying that. We never bothered to understand our structural and functional genes. So now I divided my topic into these three topics. One is on whole genome sequence, second one is on transcriptome profiling and proteomics, and the third one is the genomic assisted selection. See, once if you take genomics or resources, yes, he said, see, in crop plants like maize, rice, thousands of, a lot of genomic resources is available compared to horticulture crops. It is, in fact, in a horticulture crops, there are some medicinal crops, ornamental crops where they never touched. Only the crops like tomato, potato, papaya, 4 or 5, only they have touched. Other crops they never touch. So a lot of, very, very less information is available in horticulture. So once if you see, I just went on, reviewing what are all the things which happened in horticulture. Once if you see, you can divide it into, there are a lot of papers on genetic diversity, followed by tagging of biotic and abiotic stresses, then cultivar identification, gene pyramiding, molecular assisted breeding, linkage and molecular marker maps, and to address the IPR issues. And also, there are some papers on, excuse me, hello. There are some papers on male sterility, there are some papers on sex, habit types, homoclonal variation. All these, there are some papers. Of course, not many have come into the products, but what I did was, these are of course the markers. So what I wanted to do is, first I just, listed what are all the work which is going on in ICR institutes and some universities. Then I have listed the works which is, done elsewhere. Because people think that, biotechnology, nothing is going on in India. It is only, hodgepodge work, nothing is concrete work is going on. But if I show what work is going on in ICR institutes, especially in horticulture crops, probably you will appreciate that a lot of work is going on. And once if coming to the whole genome sequence, probably you know that whole genome sequence is now just started in horticulture crops. I'll show you which crops are there. It helps many, it helps in identifying the complete set of genes. It helps in identification of disease, insect and drought resistant, even DNA forensic we can see. So a lot of advantages are there in whole genome sequence. So if you see at the horticulture, there are 29 crops which they have listed. and it is going for a like whole genome sequence. 6 already completed, as I said, potato, tomato, then papaya, watermelon and cucumber. Some 5-6 crops already it is completed. 4 are in draft and 19 are in progress, and 29 crops are like they just started whole genome sequence. In ICR, just we started mango, brinjal, pepper and cardamom. These 4 crops has been given for this 12th plan, so that by the end of another 2 years, these 4 crops whole genome sequence will be there. So maybe you ask that why, say brinjal is also very important, other crops is also very important, why we cannot go? Yes, it is very difficult to go for whole genome sequence of all the crops because the money is the criteria, so many other things. So if at all, if you want to work on a crop, if you can't go for the whole genome sequence, definitely you can, another thing is the express sequence tags, ESTs. So if you have a good ESTs, again you can do a lot of work. You can derive your own primers, you can derive your own SSRs, you can identify the gene. So again you have to look for the EST resources. There are a lot of EST resources. Again, compared to the rice or wheat or maize or some croplands, in horticulture crops, only tomato and potato has got highest, followed by some other crops. So this is the EST sequences which is available in horticulture crops. So except this solanum, the potato and tomato, all other crops, like very less is there. So you can see here. So ESTs are the one which, like as a student, as a researcher, if I don't get the whole genome sequence, you can definitely think about ESTs, download the ESTs, prepare your own in silico mining, go for SSRs and go for your identification of the gene. So second one is the transcriptome analysis, which I told, transcriptome profiling and proteomics. Again here, whenever you can't do whole genome sequence. And one more thing, good thing is this, which examines the expression of mRNAs in a given condition. Say suppose you want to work at area, to take 2 plants, one susceptible, one expressing during the fusarium resistance. So that gives a lot of information in transcriptomics, and you can see here in ICR, we have around, in horticulture, there are 17 crops and 54 triads we are working on transcriptome profiling. So probably it is a very big, like a project which recently, there is a platform called Genomic platform, where we have listed around 17 crops and 54 triads. You can see here, definitely we are, after 4 or 5 years, we will get, we can use this data for microarray, then integration of molecular and genetic linkage maps, then which accelerates the process of fine mapping, positional cloning, comparative genomic analysis and clone by clone sequencing after this, once if you go for this profile. So these are the crops, like I listed, which are the crop, the traits are targeted and institute. You can see here tomato, then potato, so many, like targets they have, they are going for a transcriptome profiling. So that once if this is targeted, you will get a lot of information, and you can definitely use that information for improving the crop. So you can see here banana, black pepper, grapes, coconut, cardamom, turmeric, and these are all, like taken up in the ICR institutes. I have not listed the universities because I did not get any information from the universities, but I had an information that which are all the crops and which are all the traits which their ICR institutes are doing. So you can see here walnut, even in orchids also, biotic stress, NRC orchid and CMV resistance, they are doing it. So that means lot of work is going on in, at least in whole genome sequence, and definitely more work is going on even in the transcriptome analysis. Then the third is, probably this is a sort of, like darling for everyone, molecular assisted selection, genomic wide selection, genomic assisted selection. It is, like terms they use. Yes, of course, this is another one area where, like you will get a lot of information, like you can go for a discovery of novel genes, you will get a superior combination of multiple alleles, then you will get a lot of information on genetic relationship, you will get association between specific genetic variants and diseases. So this is where actually, like a lot of research is going on, a lot of, theses have come out, lot of hundreds of papers have come out on molecular assisted selection. But yes, I'll tell you after 4 or 5 years, definitely I'll show you how this molecular assisted selection is going to bring out the products in future, maybe after some time. So just for a minute, I wanted to say like what in molecular assisted selection, we are interested in, like identification of this gene of interest, the marker sequence, if this is the gene of sequence. So I don't want to go into the details. So you can see here, in ICR, there are 23 crops, 78 triads, and on 12 institutes. they are working on molecular acid selection in horticulture crops. So it is a huge, project where 23 crops are there. So these are the crops, like fruit crops, there are 7 crops, vegetables, tubers, plantations, spices and other. So where the molecular acid selection, lot of projects are going on, lot of funds has been given in ICR to carry out the molecular acid selection. These are the crops, these are the institute, and try it, you can see. So the first always stands the mango, guava, banana, again lychee, grapes, coconut, then tomato, chili, brinjal, black pepper, cardamom, ginger, spices, nutmeg. You can name any crop in horticulture, mostly we are working in ICR institutes. So it is again a huge project which is going on in ICR. So I just want to go sort of glimpse with the major crops. I will not deal with all the crops. Very few crops I am just going to tell how we are working, so that as a students you will get an idea how we can really write a project for a DBT or a DST or CSIR or a UGC. See, this is in mango. We are now identified as this chalcone isomerase, flavononcin, anthocyanin, and about for the color. So these are the pathways genes which they have now cloned, and you can see here based on the pathways, we have we got a clustering, very beautiful clustering based on the color. It has come based on this gene. So this is one of the projects which is going on, and you can see here almost 387 mango cultivars diversity we did it, and so beautifully all the northeast varieties they come at one side, and all the southwest varieties they come on the other side. So that means the whatever especially SSRs which we use, it is working very well with the whether it is a diversity or identification of gene. So again, mango molecular characterization, you can see here, these are the triads for acidity, and we have already putative marker triad association we have already identified in IHR. So again, this information will be useful for a breeder, next to breed, whenever the varieties he wants to breed for a lower acidity or a higher acidity, whatever it may be. So in fact, mango DNA fingerprinting for almost 364 varieties. Yes, even in fact, many will ask what is the use. You might have done DNA fingerprinting. I will tell you how it is commercially we exploited it. See, recently the Alfonso growers from Shumaga, they came and got it printed their Alfonso from us, and because the Alfonso from Maharashtra only used to get export because they call it as Apus. Once if it is a Alfonso from Shumaga, they never used to accept. But now we gave a fingerprinting that Alfonso of Shumaga and Alfonso of Maharashtra and Alfonso of Tamil Nadu is same. So with this, certificate, they could able to get the export of Alfonso. So that is a sort of commercial interest, and plus IPR issues. See, now we have fingerprinted almost all Indian varieties. Probably nobody can take now our varieties, mango varieties. So coming to the guava, yes, the pink pulp is very important, soft seed is very important. So we did a lot of work on guava, and especially I did work on this, and we could able to do some QTLs and linkage map on QTLs. We could able to find out 8 or 9 QTLs for the soft seed and pink pulp. So you can imagine it in a traditional breeding, it is very difficult to get a soft seed and a pink flesh, because always the soft seed and a pink flesh is link characters, hard seed and a pink flesh. So now based on our biotechnological QTL mapping, we could able to now we can easily break this association linkage. This is how this will be helpful in future for a breeders. Then coming to the even candidate gene, see, we thought that the Allahabad Safeda white has got a different gene, and the pink varieties, they got a different gene. But now using this genome, using this candidate gene approach, we just found that it is carotenoid pathway, a single indel is responsible for the color. Alfonso, this Allahabad Safeda is becoming white just because that a chalcone synthase is not working. So this is how, it helps the breeder. Otherwise, the genes are present in both the pink genes are there even in Allahabad Safeda also. But just one indel is changing the color. So banana, in fact, lot of work is going on, especially in our NRC banana in Trichy. They did a lot of work. In fact, I borrowed the slides from them. They have, so you can see here, cryopreservation. See, now this is the time we are now preserving lot of varieties. After cryopreserving, they wanted to check the stability, and they found that stability is good with the cryopreserve. So this simple give message that you can go for a cryopreservation of any variety without any problem in banana. So this is a sort of commercial application which I wanted to tell, Then fidelity, you can see here using these SSR markers we can identify the fidelity, because especially in the companies. So whenever this bananas they sell, whenever a farmer is not growing properly, immediately you will say, sir, he has given a different plants, it is not proper and all. So now with this, few SSRs, I can identify whether this variety what he has given is correct or not using this fidelity test. Then even there are some work is going on in IHR and NRCPB on drought resistant bananas. Then fusarium wilt, you know that there are some one or two varieties which are almost become extinct because of the fusarium wilt. Now a lot of SSR markers have been identified for resistance. Then around 147 SSRs we have used, almost 8 defensive genes also we identified for the fusarium wilt. We found that there is one Calcutta 4 which is resistant, now can be used for breeding resistance for fusarium. And Bunchita, of course, now they have identified a few markers. Then you can see here Cavendish. Within Cavendish, lot of groupings they did on diversity, on Bulbasiana. So even the NRCPB and IHR together, they are doing this banana draft genome. So maybe within 1 or 2 years, complete draft genome of Bulbasiana may come so that it can be used for banana breeders. Then coming to the diagnostic kits, see now diagnostic kits is very, very important because the future, like just natal markers, diagnostic kits also are very, very important, especially in quality planting material. In fact, this is the kit which we made in IHR. The Maharashtra government has purchased this kit by paying almost 8 lakh rupees. See here, very simple is in citrus, rough lemon and Rangpur lime they use as a rootstock. So many times, the nursery fellow, when they don't get Rangpur lime and rough lemon, you use a variety called Galgal. So that after 5 years, that Galgal, the one which the plant which is grafted on Galgal will collapse. So the government of Maharashtra has asked us to give a sort of, a diagnostic kit. We made a scar diagnostic kit, we patented it also, we commercialized it also. What I wanted to tell is, in future, these are the diagnostic kits, especially when the Nursery Act is passed in Parliament in maybe shortly. So once if that is there, diagnostic kit to identify spurious planting material will be in a big hit. So for all the planting material, this type of diagnostic kits is required and which is going on. So you can see yearly, we have just multiplex PCR we did with these 3 primers. I can identify whether he has grafted it on Rangpur lime, whether he has grafted it on rough lemon or Galgal. Very easily we can identify. So that now the Maharashtra government made it a sort of certification that each and every plant has to be certified. Even if it is 20 rupees per plant extra, there is no problem. Because after 5 years, the plant which is grafted on Galgal will collapse. So that is why they want this, Then pomegranate, yes, it is now a big hit, like a big problem because of Xanthomonas wilt. It has become a big problem. In fact, lot of men and material is going for a identification of resistance source for the pomegranate. So we are doing lot of marker development work. We only developed lot of SSRs because absolutely there is no SSRs. Using ESPs of other crops, we developed SSRs. In fact, some small linkage map also we did. So maybe by another 1 or 2 years, because we are using some wild varieties called Darwin Nana, we could able to identify the genes responsible for the Xanthomonas wilt. So jackfruit, of course, one of our students, they did some, markers for the cracking types. See how, once if you have this type of information, you can just take a leaf of a jack and say that this is a cracking type, just don't take it. So this is how, commercially you can use it. Then in passion fruit, there is a diversity studies. Then, yeah, this is where I am telling. See, tomato, lot of work has been done in IHR. So we have used, TY1, TY2, this is TLCB resistant, 3 genes. We bought it at one parent and we could able to bring out a variety called Arka Rakshak. You can see here, 3, TY1, TY2, TY3, 3 different strains of TLCB plus bacterial wilt plus early blight. All 3 genes has been pyramided using the molecular markers and our Arka Rakshak variety has been released. In fact, I am telling you, these parents have been sold at least for 10 companies because ICR is not an exclusive. We sold it for companies anywhere ranging from 5 to 20 lakhs for each parent. So in fact, now almost 12 countries, they wanted these parents because so far this is the first triple hybrid in tomato with TLCB, bacterial with an early blight. Then chili, a lot of work on capsaicin. You can see here almost 8 QTLs were identified by Indian Institute of Vegetable Research, Varanasi. So again, for the first time, 2 QTLs were determined for the Valorecin also. Then again Phytophthora, we could able to identify 2 genes so that we can utilize it. Now we are breeding chili resistant to Phytophthora. Then lot of diversity work is going on in onion, then garlic also. Then you can see here rust resistance. Earlier we thought that we have a resistant variety called Arca Anub, and I mean susceptible, very good variety, and we have a resistant variety IC 525260. So you can see here Arca Anub, earlier we thought that it is resistant only because of one gene. Now after molecular markers we found that there are 2 genes in that, UR6 and UR4, and IHR34 has got UR3. So now we are combining UR6 and UR3, and now we have almost come out with a variety. So this is an advantage of like molecular markers. People thought that it is just only for a paper, like people are using molecular markers. So maybe within 1 year this variety is released with 3 genes on the background like UR3, UR6 and UR4. So again in peas, lot of work is on there on powdery mildew. We could able to identify 12 scars with the 5 SSRs, ER1, ER2, ER3. We came out with the powdery mildew resistance. Then in Bendi, in Vokra, in fact absolutely there was no SSRs at all. We could able to do our SSRs. We developed around 100 SSRs, and you can see here around 5 companies they have purchased SSR. Probably this is the first time that the SSR markers are purchased by the companies. Never, because earlier like they never used to publish. Even now like we have commercialized it first time. The Vokra SSRs, it's around 5 companies they have taken it. For each SSR we have charged some amount. So onion male sterility, again we could able to identify some mitochondrial warpace for the onion male sterility, so that we can utilize it for breeding. Then excellent work, probably morning you might have heard about the chief guest, our Chakravarti. He is the one, they did a lot of work on potato. They see almost R genes for late blight. They could able to bring all the 3 R genes together for a hybrid. You can see here late blight, then potato virus, then PCN genes in mass. They could able to bring it to hybrids. So you can see here late blight, then potato cyst nematode, potato virus B, PRLB, all they are just pyramiding all these genes using molecular markers. So like in another 1 or 2 years, at least 4 to 5 varieties, because these are all breeders with the molecular markers. There is no problem like unlike genetic engineering where the problem is there. We could able to release a variety for pyramiding 2 potato cyst nematodes resistant to resistance. And even they are going to release a sort of triplex parental line using the molecular marker. So of course coconut, you have one more paper by Niral. They could able to identify the markers for tall and dwarf. So nice. Otherwise you have to to see whether it is a tall or dwarf for 4 years. So with markers we could able to identify. Even we validated it, whether it is a tall or dwarf. Immediately after breeding you can see whether this is going to be a tall or dwarf, so that we can utilize it. Then even the hybrid purity. See, this is where again nowadays even farmers are becoming very more knowledgeable. So whenever if you say that this is a hybrid and give it to him, if it is not a hybrid, then he will sue you. The chance like America in future, definitely a farmer is going to sue, because it is going to give yield after 4 years. So in fact your hybrid purity is one of the biggest thing which in future we have to think about it. So using our molecular markers we can easily find out whether it is a hybrid, pure hybrid or not. You can see here for pepper marker wilt, it is there, like wilt resistant and wilt susceptible. Then cutted disease for small cardamom, again we identified. Then microsatellite markers. You can see here beautifully cardamom, all the morphological characters with molecular markers it is there, so that once you can utilize. Fidelity. See, this is one thing. Now like papaya seeds they have grinded, like in pepper they mix. Easily like fidelity test we can identify. This is very important, because accreditation now is there, so that once if you identify that papaya is mixed in that sample, the export will be rejected. So this is the one which like in future a lot of fidelity tests will come. So in fact in Anthurium also we have developed some linkage map for Xanthomonas resistant and scent. So chrysanthemum diversity studies are there. You can see Neelima and mutant easily they keep at one place. So beautifully. Then in China aster we did some work. Then lot of like almost we fingerprinted 394 varieties. Sir, just 2 minutes I finish. So So I don't want to go work other than India. Probably you now understood that there are enough work is going on in ICR institutes, enough work is going on in the universities. Only thing now we have to utilize it for a product to bring. See, there are a lot of other work is like you can see fusarium wilt. Since so many genes are available, it is now the students has to take these genes and work. Tomato for fusarium wilt, again tomato for fusarium wilt, powdery mildew, you can see hundreds of genes are available now. So only thing we have to utilize it. Grapes, again scar marker for powdery mildew, peas powdery mildew, peas, then rose powdery mildew resistant genes. See, they are all converted into scar markers. Now it is easy to use it. Phytophthora, again you see number of genes which are available there. Then late blight potato, it's already I told, pepper, phytophthora, it is there. Anthracnose, again you see so many genes are there available. So again beans, anthracnose, it is available. Strawberry, anthracnose, gladiolus, fusarium. These are all the genes which are available. Lady's finger, male sterility, then anthurium for colors, habit, everything is, genes are available. So it is now, like the era of molecular markers has already begun, already the products are coming out. Probably it is now the students has to utilize it, the researchers has to utilize these markers, and you have to pyramid those genes to bring out the product. Probably this is last slide which I want to put it is, say earlier, whenever we want to develop a markers, they used to say, like population is required. Now, you know that now association mapping has come. Dr. Vinod is here, you must be telling more about it. Now using germplasm, now you can identify the marker with the triad. So this is another association mapping which now we are doing it in guava, cashew, then citrus, pomegranate, cashew, jack and all, we are using it, the association mapping. So this is the last slide. What I want to tell is, see, only biotechnologists cannot do anything. Only breeders also cannot do anything. So it is the both, the phenotyping should be done by a breeder and genotyping should be done by a biotechnologist. If both of them synergistically, if they come together, definitely we can improve the crop, we can bring the changes, we can definitely bring the revolution in the horticulture or any crop. So normally what will happen, biotechnologists say I am doing separately, breeders say I am doing separate, definitely it is not. It is only both of them should synergistic. Thank you for your attention.
I have only one question.
Yeah.
You projected about the male sterility in okra.
Yes, sir.
And that gene is?
Sir, it is gene.
Gene. And you have tagged it?
Yes, we have tagged it. We sold those lines also for almost 10 companies.
Well, so you do with sequence details?
No, we don't give that sequence details.
But my friend is very keen.
That you can buy from us actually. If you want male sterile lines, you can buy. It is very cheap.
No, markets, primers.
Yeah. No, SSR companies in what is that?
Identification
yeah,
of.
yeah, that we are giving. That sequence we give. That sequence we give. Not male sterile. SSR markers which we are selling to okra markers, we give sequence. There is no problem. Sir.
So, excuse me, one more here. Dr.
Yeah.
So, you are shown a slide on banana cryopreservation.
Yeah.
So, whether you preserve the seeds or the
no,
clones?
they preserved the seeds.
Seeds. Okay.
Then they raised it and they found that we can cryopreserve it after cryopreserve after so many years, nothing has changed.
Okay, fine.
It is seeds as well as
that's
explants. Explants also. Explants for 2 years.
2 years.
Yeah, fidelity is still there.
Okay.
There is no change.
Oh, fine. Thank you.
Yeah, very comprehensive one. Nice review. Thank you.
Thank you very much for.
He is the principal scientist of wheat breeding station located in Wellington, the Nilgiris. He is helping India nation to grow a lot of off-season nursery and a facilitator to much of the wheat breeding program. We welcome Dr. Sivasamy. Please.
Thank you. Respected Chairman, delegates from across the country, my staff colleagues, scientists from this institution, organizers, and student friends. I am very thankful to the organizers for giving a chance. I am being an alumni of TNAO. I feel honored for giving the chance to make a presentation. The global wheat production has made a record yield of 717 million tons last year, and it's expected to be in the order of 722 million tons next year, expected to be. But if you just look at the production levels, China stands first with a production level of 126, but the yield become almost static. India, although made considerable progress, particularly last year we made 95.91 million tonnes as a record yield. However, it's a slight almost 1 million tonnes more, we could able to produce more. But the situation in other countries like in USA, it is falling, and also in Australia, Turkey. But the actual reality is, in the future, no one is going to export wheat. Even if we have a shortage in India, and we cannot import. Although the largest exporter is Russia, but we cannot buy from Russia because it's a red grain. They prefer only red grain, but we prefer amber grain. So the only situation is India has to produce its own food in the future as well, and expect around 120 million tonnes in the turn of 2020. So we have to make almost 2 million tonnes annually incremental yield, so that we can reach that level. The Indian wheat production, you can see the gradual increase over the year, and now we have reached the record yield level of almost the productivity of 3 plus, just above the global average. The constraints in wheat production mainly of 3 rusts, particularly black rust, brown rust, yellow rust, and now the emerging problem or the powdery mildew on the blight. But the leaf rust caused by Puccinia triticine, but causing about 20 to 40% yield loss. It survives both in cooler climate and also in